Senhoras e senhores, bom dia. Ladies and gentlemen, good morning. Welcome to the 7th World Palm Symposium. On behalf of the organizing committee composed by myself, Rita Portela, Thais Emílio, and Alexandra Pires, and the scientific committee of the event, it's a great pleasure to receive you all in this opening ceremony of the World Palm Symposium. The World Palm Symposium happens every 5 years, and this edition, Palms 2021, is the 7th in a series following Colombia 2015 and previous edition in Montpellier, London, New York, Miami, and its first edition in Ithaca, 1987. We organized an event full of opportunities for palm researchers to interact scientifically, socially, and to build a stronger palm research community. We are happy to have the participation of more than 70 senior and early career professionals and more than 60 students from 75 cities, 26 countries, and 4 continents, and proud to make this event really global. In this edition, our Palm Symposium meeting will be virtual, but we hope that we can be back in the presential format in the next edition. Meanwhile, let's enjoy the great benefits of making content and participation more democratic offered by the online format. We worked together with the scientific committee to prepare a rich and diverse program to this event. We wish you to enjoy it and take each and every opportunity to learn from the wonderful community that's together here for this event. Welcome to Palms 2021. We appreciate the financial support given by the Fundação de Amparo à Pesquisa do Estado do Rio de Janeiro, FAPERJ, International Palm Society, and Montgomery Botanical Center. We also thank for the funding offered by some professionals to support students' participation. They are Larry Norbeck, Patrick Griffin, Joanna Tucker Lima, and Thomas Courvier. Before we move in the program, I would like to make 3 announcements. First, that the 7th World Palm Symposium will grant a Best Presentation and Best Poster Award. The chairs of each section will nominate a candidate for each category. The decision will be made by the scientific committee based on merit and popular vote. So please rate all the posters and presentation you watch, so your opinion could count. Results of the 7th World Palm Symposium Prize will be announced in the closing ceremony. Stay tuned. Second, we need to start to think about the next edition, hopefully in 2025. It will be wonderful if you can already have some candidates to host the next meeting. If you want to host the next World Palm Symposium, get in touch with us, so we can share our experience and also arrange some time for you to present your candidature in the closing ceremony. Third, in order to make our experience even more complete, fun, and with the feeling of being a face-to-face event, we will have a talent show to break the ice. We will mail you to complete a form and check what talents we have here. With this, we would like to thank you for your attention and move to our first keynote talk, which we are very much looking forward. It's a great pleasure to present Professor Rosane Collevatti. Rosane is an evolutionary geneticist and professor of the Federal University of Goiás, Brazil. She has PhD in ecology from the University of Brasilia. Professor Collevatti focused her research on population genetics and genomics of neotropical trees and palms, with particular emphasis on geographic patterns in neutral, quantitative, adaptive variation in cerrado species. She also focuses her work in community phylogeny and community assembly in the Neotropics. The title of her talk is Bridging Population Genetics, Genomics, and Macroecology to Understand the Evolution of Neotropical Palms. The duration of the talk will be around 40 minutes, and the last 20 minutes will be devoted to a question and answer section. You will be able to ask questions during the talk in the Q&A box in the bottom of your Zoom webinar screen. Please note that only questions asked in the Q&A box will be collected and directed to Rosane. But feel free to make comments and interact in the chat box during the Q&A section. Rosane, thank you so much for accepting to Open Palms 2021. The word is with you now.
Okay, Rita, thank you. Thank you, Thais, and all the committee for this great opportunity to present our work in the World Palm Symposium. Today I will present our study focusing on the evolution of palm species, and basically using this framework coupling population genetics and genomics and macroecology to understand how demographical dynamics and natural selection shape the evolution of neotropical palm species. Thank you very much for you all that is watching my presentation. I will now share my my slides, my video. Just a minute. Okay. Rita, please let me know if anything wrong happens with my slides, okay? Is it working now? Okay.
Yes, it's working.
Okay, you can see my slides.
Yes.
Yes, okay.
Perfect.
Okay, so the work that I will present today is part of mainly two projects that I and Christine Bacon are PAs, and these projects are funded by Brazilian and Sweden agencies. And I would like to acknowledge all researchers and mainly the PhD students that are working hardly in these projects to achieve all the results that I will show to you today. And today we'll focus mainly on two species with contrasting life histories. One is Acrocomia coleata, and the other one is Mauritia flexuosa. While Acrocomia belongs to a species-rich tribe, Cucosi, in Neotropics, Mauritia belongs to a species poor in Neotropics, Lepidocari. I will also show the results of two other species, Acrocomia totai and tumescens, that have narrow geographic distribution because they both hybridize with Acrocomia coleata, as I will show in the following slides. Okay, so although both species are widespread in the Neotropics, Mauritia flexuosa is widespread in South America and has cis-Andean distribution, as you can see here, and has high local abundance, forming these monospecific stands in swamping habitats that we call here in Brazil veredas, in Colombia and Venezuela morichales, and in Peru aguajales. The other sister species of Mauritia is Mauritia carana, and contrasting with flexuosa, this sister species has a narrow distribution in the North South America, and it's not abundant as flexuosa. We still have no results on Mauritia carana, but we really expect to have results in the near future. So I will not present anything about Mauritia carana today. Acrocomia coleata is also widespread in the Neotropics from the Northeast Brazil here in red, the red cycles here, and towards the Mesoamerica. And it's wider geographic distribution compared to Mauritia flexuosa. However, it's not a hyperdominant species and not as abundant, local abundant as Mauritia flexuosa. And although it's widespread, it occurs mainly in seasonally dry ecosystems like Cerrado, savannas, Brazilian savannas, and seasonally dry forest in Atlantic forest and in Argentina and Bolivia, Paraguay, and in seasonally dry forests in Mesoamerica and Llanos. The other 7 species of Acrocomia have narrow distribution compared to Aculeata, like, for example, Acrocomia totai, that we'll talk about today, in the South Brazil, Argentina, Bolivia, and Paraguay. And Acrocomia intumescens is an endemic species in the Northeast Brazil, occurring only on a very narrow distribution in Atlantic forest and also in islands of forests inside Caatinga, forests that we call brejos forest. They occur in only on those 2 places. It's also important to note that Acrocomia culeata distribution is parapatric to Acrocomia intumescens here in the Northeast, and parapatric to totai, and with some overlap in the south here, in South Brazil. The fossil record shows that populations of Mauritia flexuosa expanded and retracted during the Quaternary. So during the glacial cycles here, the pollen shows a lower frequency of Mauritia in the pollen fossil record during the glacial periods and a higher frequency after the last glaciation. So the fossil record shows an expansion of geographic distribution from the last glacial cycle to the present. And this dynamics in Mauritia geographical range is also evinced in the ecological niche modeling. So ecological niche modeling shows a narrow distribution of Mauritia flexuosa in the last glacial cycle, and this distribution expanded from the LGM across the time to present day. So we found an agreement of the climatic refugium, which is areas of climatic stability through time since the LGM. And there is an agreement of this climatic refugium and the pollen fossil record that shows an absence of a lower frequency of Mauritia pollen during the dry and cold periods compared to the wet and hotter periods of the last glacial cycle. Thus, Mauritia populations may have less in suitable sites during glacial times, which we call multiple refugium pattern of demographic dynamics. And these demographic dynamics may explain the genetic database on microsatellite loci that show high differentiation in a more broad scale. So as we see here, we have a high differentiation among populations from different ecosystems like Amazon, Llanos, savannas in the central Brazil, and also in a more regional scale, such as river basins, such as Amazon Basin, Araguaia, Tocantins, Paraná, Orinoco, and North Atlantic. Also, lineages from Amazon are more ancient than lineages in Cerrado, and also lineages in Amazon Basin, river basin, are more ancient than the river basins in Cerrado. So Amazon Basin, and then Araguaia, Tocantins, followed by the other river basins. And this pattern matches the pattern in migration based on coalescence model, and also the demographic dynamics during the Quaternary with populations remaining in microclimate refuge during the glacial times. Neutral SNP loci also recovered this pattern of high differentiation among biomes or ecosystems, as you like, such as savannas and Amazon and llanos, and also among river basins. In simulations of demographic dynamics, based on different genetic data such as microsatellites, chloroplast sequencing, and neutral SNP loci, recovered the microclimatic multiple refuge hypothesis during the LGM as the most likely scenario to explain variation in genetic parameters among populations of Mauritia when compared to other scenarios such as population stability or population expansion or retraction. Unfortunately, different from Mauritia, Acrocomia's fossil record is sparse, and the dry ecosystem that Acrocomia coleata occurs does not favor the preservation of fossil. And we still need to take a look at the spatial and temporal pattern in the variation of pollen abundance to verify whether it matches with ecological niche modeling, which we are going to do now. But the most ancient record dates from the beginning of the Holocene in a site here in Amazon River Basin. But the ecological niche modeling recovered broader geographic distribution of Aculeata in the last glaciation, contrasting to Mauritia. So Aculeata had a broader geographic distribution than present day, and the geographic distribution retract during the time. It also contrasts to the other species, Acrocomia intumescens and totai, which had the opposite pattern. So the geographic distribution retract during the last glacial maximum and expanded through time until present day. This expansion in Acrocomia coleata, this former expansion during the LGM, may explain partially the contact zones that we observe in the genetic data between Aculeata and the other two species. Because nowadays, in the present day, the niches, the climatic niches of the three species are different, and we found lower niche, climatic niche overlap than expected by chance between pairs of species. However, genetic data based on, for example, sequencing of nuclear genes in intumescens, Aculeata, and totai show introgression of genes of intumescens in Aculeata, like this one here, this gray haplotype here, and also Aculeata in totai, like here, this blue. Okay, and also we find incomplete lineage sorting. This haplotype here can be found, which is an ancestral haplotype, can be found in all three species, like here, and here, here, and here, in the three species. Several individuals of Acrocomia totai phenotype are indeed F1 hybrids between Aculeata and totai. Like here, they have a phenotype of a totai, but they are F1 hybrids, showing ongoing introgression between these species of in nuclear genome, and nuclear genome of totai into Aculeata and vice versa. So although we have a parapatric distribution, there is an ongoing introgression between these two species. the same evidence of hybridization was recovered using neutral nucleoside. We find high differentiation among populations. For example, here in Aculeata, we have high difference among, for example, Atlantic forest, unseasonally dry forest, and savannas. But a high proportion of this difference in neutral loci is due to isolation by distance. For instance, in Aculeata, 32% of the difference between a pair of populations is due to geographic distance, while in tumescens here, 82% is due to isolation by distance, which is a lot. Well, and how adaptive variation can explain the spatial distribution and evolution of Aculeata in Mauritia flexuosa? On one hand, we have Mauritia flexuosa, a hyperdominant species that is widely distributed in different ecosystems, such as rainforests and seasonally dry forests, and but always associated with swamp habitats. On the other hand, we have Aculeata, which also has a broad distribution, but always in seasonally dry ecosystems. But for both the species, we found nucleosi with selection signal in genes related to both basic metabolism and response to abiotic and biotic stress. However, in Mauritia populations, in contrasting climate and soil conditions, we found only small differences in allele frequencies for most nucleosi, showing a potential fixation or quasi-fixation of one allele in almost all populations, while in Aculeata, populations have higher differences in allele frequencies in contrasting environments. As we can see here in these maps, for Mauritia flexuosa, we see small differences in allele frequencies here and here and here, with one allele fixed or almost fixed in populations in different ecosystems, the same allele. So we have an evidence of selective sweeps. And here, for only a few loci, we have evidence of adaptive selection for example, temperature in Orinoco or in Araguaia-Tocantins river basin. But for Acrocomia culeata, several SNPs affecting genes related to abiotic stress have signal of local adaptation, showing high difference in allele frequency in different ecosystems. For example, here and here, we have a different allele here, almost fixed or with high frequency in the transition between Cerrado and Amazon, with high topsoil grave in the soil, contrasting with the other populations with one allele fixed. And for other climatic or soil variables, we can find different patterns. For example, here, for topsoil sodicity, which is higher in Atlantic forest, we find the opposite. Atlantic forest has a different allele fixed, contrasting to the other populations that have one other allele almost fixed or fixed. And for temperature, we found several loci showing adaptation with climate pattern following the increase in temperature from the South Brazil towards the northeast, like here, these three examples. Just as curiosity, I will also show Acrocomia tumescens, which is very, very interesting because we have a very different frequency distribution in loci under selection for precipitation. So in this species, the local adaptation to different precipitation regimes may shape population genetic diversity and this spatial pattern in allele frequency. So populations in island, in Regius forest, in semi-arid climate with lower precipitation, had higher frequency of one allele contrasting with an alternative allele that has higher frequency in the coast in Atlantic forest, like here, and another example here. We found no evidence of local adaptation in Acrocomia totale. Okay, taking all together, both species are widely distributed, Aculeata only on seasonally dry forest ecosystems, and Mauritia flexuosa in seasonally dry and wet ecosystem, but highly adapted to swampy soils. They both show high variation among ecosystems in neutral loci. Populations are structured broadly in both by ecosystems or biomes, but also regionally. In the case of Aculeata, the high variation in, among biomes may be explained by isolation by distance, and in Mauritia, variation is structured by river basin, mostly likely due to its dependency to water courses or swamps. While Mauritia had a retraction in geographic range during the glacial times or periods, and then an expansion after glaciation, Aculeata had the opposite. It had a wild geographic range in the LGM, and then at present day, a narrow distribution, the distribution contract, following the pattern of expansion of dry ecosystems during the glacial times in the Neotropics. In Mauritia, we hypothesize that adaptative selective sweeps related to adaptation to swamp environments may have shaped the spatial pattern of genetic diversity, leading to high similarity in allele frequency among populations from different ecosystems. So selection together with migration led to the spread of alleles with higher fitness among populations from different ecosystems and promoted this evolutionary success of Mauritia in swampy microhabitats. In Acrocomia aculeata, in turn, local adaptation to soil conditions shaped the spatial pattern in allele distribution, with different alleles favored in populations with higher grave concentration or sodicity. On the other hand, adaptation to temperatures, to climatic conditions with gradient pattern led to climb in allele frequencies in loci under selection. But it's not all. We also investigated whether human domestication may explain the hyperdominance of Mauritia flexuosa and the spatial pattern in genetic diversity. We predicted that if the pre-Columbian domesticated Mauritia collect seeds to grow Mauritia nearby the villages, genetic diversity is better explained by distance from the ecological site rather than population suitability or stability through time. Also, because domestication may encompass the selection of seeds from just some individuals with large seeds and thicker mesocarp, genetic diversity will be reduced in the long term. Thus, genetic diversity in populations nearby ecological sites is lower than populations far from archaeological sites. To do this, we compiled genetic data from nearly 60 populations across the geographic distribution of Mauritia. We compiled data from archaeological sites and polyfossil record. From the polyfossil records, we compiled the relative abundance of Mauritia through time. So we have slices of time with the relative abundance, like here, and the spatial and temporal pattern of variation. And then we modeled the genetic diversity in relation to the distance to the archaeological site, the stability and abundance in polyfossil record, and the climatic suitability and stability based on ecological niche modeling. Models with climatic or fossil stability or the new models explained better the variation in genetic diversity among populations of Mauritia than models with only the distance to archaeological sites. And also we found no differences in genetic diversity between populations near and far from archaeological sites, meaning that demographic history led a strong signal in populations, and it's not possible to detach the effect of domestication from the effect of demographical history or demographic dynamics. Thus, we believe that if pre-Columbians domesticated or somehow started to select and grow Mauritia in their gardens or nearby their villages, the genetic signal is weaker than the signal from the demographic dynamics caused by the Quaternary climate changes. And that's all. Thank you for your attention and for your patience, and then I'm available to answer all your questions and doubts. Thank you very much.
Thank you, Rosane, very much for your wonderful talk. Now we are going to, Rosane, do you prefer that I read the questions or you read the questions? Because we have here in Q&A some questions already. What do you prefer?
Okay, Q&A, let me see. Okay, I can access here. Well, yeah. If you select questions, you can ask me, and I also can take a look here to see if we have other interesting questions, but you can.
Okay. So the first question that we have is from Monica Moraes, that's a chair of one of our section. She said, more than a question, this is a remark. Mauritia flexuosa reached its distribution to central Bolivia, not only in the northern pattern with Brazil.
Yes, because savannas also is distributed in Bolivia, Paraguay, and so Mauritia also follows this pattern of distribution. Yeah.
Yes. Okay, the other question is from André Silva Pinedo. He asked, since there is a genetic divergence among populations from different regions, do you believe that we can delineate other species, subspecies, or variates in Mauritia flexuosa and Acrocomia culeata?
Well, I'm not a botanist, so I think without botanical evidence with morphological and such as flowering differences, I cannot say anything about that. So I think genetics can give support and evidence to start a work on botanics to propose new species, but not conclude only with genetic data that we have a different species. I don't think we have enough evidence from genetics, only from genetics, to say that they are different species. But I think it's a start, you know. Given this genetic evidence, we can, together with specialists in palms, we can start to study that and see if we really can propose another species.
Okay, thank you. And the next one is from Mauro Galetti. He said, great talk, Rosane. Acrocomia crispa is endemic from Cuba and Acrocomia media from Puerto Rico. Do you know anything about the evolution of these species? How related they are from Acrocomia culeata?
Hi, Mauro. Thank you for the question. No, unfortunately not. We don't have samples from those species, but if anyone have and would like to study with us, we are really aiming to expand our work and collaboration to work with more species. So unfortunately not.
The next question is from A.G. Harris. In Acrocomia, you are attributing shared alleles, H6, green, to hybridization and presumably secondary contact. But how have you ruled out that speciation is ongoing, such as across ecological gradients and that what you are observing a stage of incomplete lineage sorting.
Okay, Harris, H6 I attributed to incomplete lineage sorting because H6 is a more ancient haplotype. So the evidence is that it's incomplete lineage sorting. The other we are attributing to hybridization. Okay.
Okay. Okay. And another question from Monica Moraes. Is that so that Acrocomia totai is expanding its distribution in a more diverse ecological areas, such as seasonal subandean forests up to 700 meters elevation?
I don't have this information, but I believe so, because we are observing in several species, animals and plants, for example, anuram, the expansion of distribution to the south, and because of climate changes, we believe. And so it's not impossible that Acrocomia totai is expanding its distribution either.
Okay. And another one here is from Shui Junji. What molecular marker from SNP are AD?
Oh no, it was not RAD, Shu Junje. I hope I had pronounced correctly. It was sequence capture. We use probes developed for palms, and we performed sequence capture, target sequencing.
Okay. And one here from Thomas Curver. Thanks for this talk, Rosane, about the suggested domestication of Mauritia. What morphological traits do you think pre-Columbian societies would have selected? Are there any morphological difference between populations that you have observed? At fruit level, maybe this would also be a strong sign of selection as suggested.
Hi, Thomas, thank you for your question. Yeah, we have to investigate these quantitative traits, such as fruit characters. I believe they were selecting for seeds for mesocarp, thicker mesocarp that they use for food. And so, you know, it's we can believe that they were selecting for this. Maybe smaller palms to be easier to collect the seeds, and maybe plants that were flowering earlier because they did not have to wait a long time to collect the seeds. But we have to study this better, and I think this is a next step. We really need students to do that, and this would be great. I have not observed any difference, great difference among populations that I collected. I can see that in some populations in savanna have smaller seeds, but this is only empirical, it's not experiment, so I can say that for sure. And I can also observe this in other species either, like Cariocar brasiliense and other species, and I believe this is soil variation and not really selection, but you know, we have to study that.
Okay. And do we have from Cynthia Freitas? Thank you for this great talk, Rosane. What are your advice for young students that want to develop projects with such an amazing sampling effort, but face short fundings?
Okay. Hi, Cynthia. Thank you for this question. This is a very interesting question because I really started this work, the work with Mauritia. I really started when I have just finished my PhD in 2000, and I really have short funding, no funding at all. So I started to collect in populations that I could, and I also started to ask friends that were working nearby places with Mauritia to collect. So when I could gather a lot of samples, I started to work with that. And Natácia Lima performed her work in phylogeography of Mauritia. And then I was acquainted to Christine Bacon. And so she was also in the beginning of her career trying to get samples from Mauritia. So we put all together and then we start to work together. So I think collaboration is everything in science, and we can do better. You saw in my slides, I have a lot of students, a lot of collaboration. And so this huge project is only possible collaborating with a lot of people. And I think it's the way.
Yes, and I think this symposium is a great opportunity to do this, to collaborate with other palm specialists, to exchange knowledge and also materials.
Yeah, great, because, you know, like I said, we want to work with Mauritia Carana. Does anyone there have samples or want to sample and gather this great group, you know, and also Acrocomia, samples from Acrocomia populations of Acrocomia. So, for example, it would be great to have populations of Acrocomia coleata from Mesoamerica, but it's really difficult to us to access populations there, and it's too expensive. You have no funding now in Brazil, so only with collaboration we can really perform this huge work.
Yes, and just to highlight, we have a lounge area on the platform that we have a network room where you can exchange knowledge and maybe try to exchange materials from this community, and also a message board that we can talk and the chat there. So just to highlight it. Another question from Cynthia Rosane, a super cool student on Pre-Columbian manipulation. Don't you think that that the fact that Mauritia is associated with water might mean that the people did not have to manipulate Mauritia, since I believe that they had to manipulate other species to bring them closer to the water and their settlements, which were already near the water?
Yes, that's my feeling. And we wrote that, Cynthia, in the microsatellites paper. Remember that you participated? Exactly that. I believe that. If you have the villages are nearby water courses, Mauritia populations are nearby water courses. Why have all this work to gather seeds and transport seeds and plant seeds if you have the plants there in water, you know? Also, seeds of Mauritia are very difficult to germinate, to grow. So I believe, I don't believe in domestication in Mauritia, but you know, it's science, you don't have to believe. We have to prove. So we are pursuing that. But my feeling is that why domesticate a plant that is exactly on where you were going, you know, where you're establishing your village. They are already there. I don't have to domesticate, to plant, to grow, and to take care.
Okay. Another question from Maria Teresa Pulido Silva. Very interesting. Our studies show that rivers can affect the gene flow. Do you found or research something related with how the rivers affect the gene flow? Please check the paper. She put the webpage by Nilo Sander.
Yes.
Yes.
Yeah, yeah. We didn't perform a local study like Sander, but in a broader scale, yes, the migration is related to rivers, and the pattern of migration recovered by Qualisend models agrees with the pattern of differentiation by river basin. But at local scale, we didn't perform this kind of study like Sanders did, yes.
Okay. And another one from Maria Julia Gropa Rodrigues. Thanks, Rosane, for the great lecture. Do you study other palm species from different types of vegetation in Brazil beyond Cerrado?
No, unfortunately not. No, I'm still focusing on these species. Yes, but I want to work with Copernicia. So we are discussing a way and funding to work with Copernicia. Natácia Lima and Christine Bacon and we are discussing that because, funding. And, we want to expand to other Acrocomia and other species, but not now.
Okay. Another one from Mauro Galetti. Last question, Rosane. Why are there so many variations in seed size in Acrocomia coleata?
Great. I would like to know either. I still don't know. Yeah. But I believe that a species that occurs in a broad geographic distribution, it's like Carioca brasiliense that I work for 20 years either. And there is a huge variation in fruit size, in seed size, and partially is, I found that, for example, for Carioca, partially is due to genetics. So during the mating, seeds that are from heterozygotes and unrelated mating are usually larger and have higher fitness. But at a broader scale, and we have to study that if it is related to soil conditions or if it is really genetics. So I really don't have this answer yet.
And Rosane, just it's not a question, but because I work with Euterpedulis, and that also has a widespread, but only in the forest and something in some places in Cerrado. But we found that there is a variation between years from the seed size for the production of the seed size. So maybe it's not so, well, for Euterpe, not fixed, but it's variation during the, it seems like related to precipitation, but we are not sure, but there is a great variation between years. So
yeah, yes, this is there is also this variation among and between seasons, between years. This some species in Cerrado shows mass seeding. And it's amazing. One year you have a huge production, and the other year you have almost non-production, so like mass seeding. So variation is really great, not just among populations, but within populations. The variation seed size is amazing, amazing.
And another question from Ju Jung Ji, I think it's correct. I hope it's correct. In Southeast Asia, there are lots of palms specialized in limestone. The phylogeographic and adaptation study is indeed very important for understanding the diversification of palms. It's just a
comment. Thank you. Yes. Thank you. Thank you.
Now is from Evandro Ferreira. For you, is the hybridization observed in Acrocomia an indicator that species are speciating, diverging now, indicating that in the past there was only one species? Does this perhaps explain the suggestion by Henderson and colleagues in 1995, the Field Guide for Palms of America, that they only support the existence of a single species in Acrocomia?
Can be. I really believe in a continuum, species as a continuum in space and time. So if you have a species with this broad geographic distribution, it's normal to think that in the edges of the distribution, you have hybridization with other species because you have an ongoing process of speciation. Yes, I think it's possible that they are still, the speciation is still ongoing, and maybe they will have enough differences in the future or not, if they continue to hybridize, but I think it's possible.
Okay, and another one from Nicholas Penafiel. Is there an evolutionary advantage for Mauritia flexuosa or any other palms to form stands versus growing in solitary?
I think there is one that I can think of, just now. It's a dioecious species. And so for a dioecious species to find mating, it's easier if you have these stands with a lot of individuals, although you can have a more mating between close relatives, but it's easier to have to mate. So this can be one evolutionary advantage. But I think it's the contrary, maybe it's so specialized in swampy habitats that it's impossible to compete with Mauritia flexuosa in these swampy habitats. And then you have a monospecific stand because of competition with other species. I think it's there, but you have to study more.
Yes. And one from Leonardo Rodrigues. Can you comment about the specific adaptation of Acrocomia intumescens to the breaches of the altitude here in the Northeast? The species occur in flatter area top. Thank you, Rosane.
Oh, great. These breaches forests are relictus from the forest when Amazon and Atlantic forest expanded and contact. And then you have these islands of Atlantic forest in the middle of Caatinga. And in specific conditions, soil conditions, like breaches, because you have, a water close to the soil, so it can sustain a forest. And Acrocomia intumescens is only found in those breaches. They are not found in Caatinga, in seasonally dry ecosystem there. So I believe they could colonize these places because of this special condition of breaches forest.
Okay. And one from Matthew Tello. Thanks for the talk. In case Mauritia is domesticated, what do you think is the difference between level of domestication through the Amazon basin, west versus east?
Yeah, well, to answer that, I have to prove domestication, that we don't have, until now, in genetics, we don't have a signal of domestication. So it's difficult to discuss different levels of domestication throughout the Amazon basin because we don't have signal of domestication. So I don't know how to answer a question because of that. We really don't have until now. We are trying to, analyze the data in different forms to find any signal of domestication, but they're not fine.
Okay. Another one from Evandro Ferreira. I believe that there is a problem with the concept of domestication used by the authors of the work on Mauritia flexuosa that you presented. Was the species really domesticated? For example, a domesticated plant like Pupunha, Bactris gazipis, when planted and abandoned without care by humans, disappears when the forest grows again. And this is also applies to other domesticated species like avocados, coffee. If left in areas where forests regenerate in the Amazon, in the case of Mauritia, it grows in wetland and does not need human care. If abandoned, it will persist. It only disappears if its habitat is destroyed.
Great.
In summary, evidence of domestication of Mauritia is not very strong in my opinion. To have it around, men just need to preserve these habitats.
Yes, I agree completely with Andrew. Yes, great comment. Yes, it's just that. Yes.
And another one from André Silva Pinedo. Some observations I made show that Mauritia flexuosa is taller in the Amazon forest than in Cerrado. Do you believe this is consequence of more humid climate of the Amazon, or is the result of human domestication?
Yes, I think it's the climate. Yes, because domestication would lead to the contrary pattern, you know, because usually human domesticate plants to be shorter because it's easier to gather the seeds and to manage the plant. So if domestication had a strong signal, I think we would find an opposite pattern. Taller species in non-domesticated populations such as in Cerrado and smaller nearby archaeological sites. Yeah.
Okay. And one from Lourdes Fallen, great talking, Rosane. In Peruvian Amazon, there are a kind of varieties of Mauritia fruits. One of them is sweeter and fleshy than others, and consequently, they are more appreciated in local markets. Do you think this fruit trait variation was modulated by pre-Columbian people or maybe by animal dispersers?
I don't believe that it was manipulated. We also find this here in Cerrado. This variation in not only in the mesocarp thickness, but also in the taste. We also found fleshly, sweeter seeds in some individuals than in others. So I don't think it was modulated because we would find higher number of individuals with this character, sweeter and fleshly seeds. We also find this in other species either. In Carioca brasiliense, we also have this huge variation in color, in size, in taste. It's very common.
Okay. So we have some comments here, Rosane, in the chat box. Just a lot of people, congratulations about the talk. And I think we do not have more questions in Q&A box.
Christine is asking for you to save the QA for the speakers to go back to you.
Okay.
If it's possible to be very nice, because you know, a lot of ideas.
Yes, we can do it and send to you. Okay. So I think we've done here.
Okay. Thank you very much. Thank everybody for the questions and contributions and the comments. And thank you, Rita and Thais and everybody for this great opportunity. Thank you very much.
Rosane, we have one last question here. Do you mind?
Yes.
We have time.
I love questions.
Yes, okay. So it's from Fabio Martinho Zamboni. Good morning. You could tell if there is evidence that there are population of Eutripia oleracea, açaizeiro, that have underwent a process of improvement by the pre-Columbian peoples for fruit production, like the pupunheira.
Okay, sorry, no, I don't know about the Eutripia oleracea. I think Clements can help you about that because he studies domestication in Amazon species, and we are also collaborating in other works, but sorry, no.
Okay. So thank you, Rosane, very much. We have a quick break now. We are going to return at 11 with a Q&A section about biogeography, macroecology, and macroevolution. So have a coffee break at home.
At home now. Thank you all. Thanks very much.
Thank you, Rosane, very much for opening Palms 2021. It was a pleasure and an honor for us to have you here.
It was an honor for me. Thank you. Bye. Bye, everybody. Thank you.
Bye-bye. See you soon at 11.